Hey guys, I'm Hannah, and I'm going to be talking about the principles of taxonomy. Taxonomy is the science of naming organisms and defining them for the purpose of arranging them into hierarchical groups. The way that organisms would be grouped together is if they have similar properties and they'd be separated from organisms that are different from them. Taxonomy can be seen as three connected but separate areas. The first one being identification, which is the process of distinguishing and isolate a group of cells descended from a single cell to determine which group it belongs to. The second one being classification, which is the process of organizing organisms into groups that are somewhat related or similar, making it easier to identify them for studies. And the last one is nomenclature, which is the system of organisms getting assigned names. Included in the taxonomy is the hierarchical order that classification categories are arranged into. If there wasn't a classification about organisms, scientists and other people wouldn't be able to communicate about them. It includes the domain, which is the very top, and it's a group of similar kingdoms. Kingdom, a group of similar divisions or phyla. Phylum, a group of similar classes. And phylum names usually end in OTA. And then classes are a group of similar orders. And class names usually end in IA. And order, a group of similar families. Order names usually end in ALES. And family, which is a group of similar genera, family names end in ACEAE. And genus, a group of similar species. And species, a collection of closely related individual or strains. And then this image shows the order of which it goes in.
Hi, I'm Maddox Poole. I'm currently doing the slide for methods of microbial identification based on genotype. So being able to identify the specific species of microorganism based off their genotypes has a massive medical significance because it allows doctors and scientists to prescribe medication that will specifically fight that microorganism. There are two main ways scientists are able to detect specific nucleotide sequences. These are through a nucleic acid amplification test and DNA probes. Nucleic acid amplification tests work according to the source I provided. The test can be used to increase the number of copies of specific nucleotide sequences that serve as identifying markers. In other words, this allows researchers to detect the sequence in samples such as body fluids, soil, food, and water. This method can be used to detect microorganisms even in extremely small amounts, including those that are usable to grow in cultures. DNA probes, according to the other source I provided, a DNA probe is a single standard piece of DNA that has been tagged with a detectable marker such as a radioisotope or a fluorescent dye. In other words, the probe is able to attach a complementary nucleotide sequence of interest, then brings that sequence and its detectable marker with it. By doing this, we can locate nucleotide sequences that then give us the characteristics for a particular species or group. Both these tests have a significant limitation though. Each amplification or probe detects only a single possibility. If an organism in question could be one of 5 different species or related groups, then 5 distinguishable amplifications are needed.
All right, what's going on, guys? I'm Zach. Today we're going to be talking about identification methods based on phenotype. So we're going to start with what are phenotyping characteristics. They are characteristics of microorganisms that aid in the process of identifying the organisms without the need of sophisticated equipment. Phenotyping makes the identification process more efficient. By knowing a few facts about the microorganism, you can narrow down what type of bacteria you're looking at. One of the most efficient ways scientists identify microorganism is through morphology, which is the size, shape, and the stain of the microorganism. So let's get into gram staining. It's the first thing that is looked at during the identification period because it is arguably the most useful. The gram stain can even give enough information to start treatment for the microorganism. And others like Streptococcus pyogenes, more tests need to be run. A series of tests are done to determine whether a microorganism is gram-positive or gram-negative. As shown in the chart on the right, it is important to do these tests because one small mistake could result in improper treatment. So as we look at the chart on the right, we got the gram stain up top. We're going to go to the left. So gram-positive coccus, you test for positive or negative for catalase. If it's positive, you run another test to get the microorganism, or if it's negative, then you already know the microorganism. So that really showcases the importance of the gram staining technique. Biochemical tests are used to discover metabolic capabilities, which is the ability to perform metabolic function of microorganisms. The presence of catalase is a common one that is used for testing. Dichotomous key is a series of alternative choices that lead to microbes identification. Because incubation periods take so long when identifying microorganisms, multiple biochemical tests are run simultaneously. Serological statistics is a type of testing that uses different antibodies to detect polysaccharides and proteins to identify organisms. There are identifying factors that can help expedite the process, such as the method of movement, flagella or pilus, or if there is a capsule or slime layer present. MALDI-TOF, it's an acronym for Matrix-Assisted Laser Desorption Ionization Time of Flight Mass Spectrometry. It's new in the field and it's revolutionizing because of its speed in identifying microorganisms. It measures the mass of the components of the organism to determine its chemical makeup. There is a beam that breaks down and ionizes the molecules and leaves the larger molecules still intact, termed soft ionization. The ions then travel into a flight tube, and the ions are separated by their mass. The mass spectrum, which is the pattern of peaks that you see here on the left in the purple graph, allows the microorganisms to be identified.
Hi. We're also going to be talking about how scientists can use rDNA to identify organisms. On top of using the two tests that we talked about earlier to identify DNA sequences, scientists can also use the sequence of ribosomal RNA genes. According to the source I provided, the nucleotide sequence of ribosomal RNA molecules, or the NA that encodes them, can be used to identify microbes. But why is this the case? Well, rRNAs are useful in microbial classification and identification because they are present in all organisms and their sequences are relatively stable. The ribosome would not function with too many mutations. However, not all regions of the ribosomal sequence are stable. Some parts have relatively few changes, but others have more variable. We can also use rDNA to identify uncultivated organisms. The vast majority of microbes cannot yet be grown in a culture. However, their DNA from those same organisms can be amplified, cloned, and sequenced, making it possible to detect and identify them.
Distinguishing different strains is important and useful, especially when tracking the sources of certain infections. There are 5 main ways of distinguishing different strains: biochemical typing, serological typing, whole genome sequencing, phage typing, and antibiogram typing. Biochemical typing, you can differentiate different species by biochemical tests. A group of strains that have a characteristic biochemical pattern is called a biovar or a biotype. Serological typing are proteins and carbohydrates that can vary among bacteria strains, and so you can be used to differentiate. A group of strains that have cell surface antigens that differ from other strains are called serovar or serotype. Whole genome sequencing. Now, scientists can detect small differences in phenotypically similar strains by using DNA sequencing and comparing the DNA of the whole genome of the different strains. Phage typing. These are the two last ones of the different strains. Now, phage typing strains can also differ in their susceptibility to bacterial phages. Now, bacterial phages are viruses that infect bacteria and often lysine them, so it destroys the bacteria. If the bacterial strain is susceptible to the specific type of phage, a clear area will form on the surface of the agar plate, just like this, where bacterial cells once were. Antibiogram typing. This differentiation is found by observing antimicrobial susceptibility patterns of antibiograms. To determine the susceptibility patterns, a culture is inoculated onto the surface of the agar. Then, using paper discs containing different antibiotics, you place them on top of the cultured bacteria. After incubation, clear zones called zones of inhibition will show around the disc, confirming if the antibiotics killed the strain, just like here as well. That is the zone of inhibition.
Hi, I'm Anji. So fungi, we're going to talk about their types, reproduction, and medical importance. So fungi are heterotrophs, meaning they can't make their own food like plants. Many are sapotrophs, which means they absorb nutrients from dead or decaying matter. Their biggest role in nature is to be a decomposer. An example of this is if a tree falls down in the forest, the fungi breaks down the wood by digesting the lichen and the cellulose. When fungi break things down, they release carbon dioxide and nitrogen compounds back into the environment. Other organisms then recycle these compounds, helping them to keep the ecosystem balanced. It's like a nice partnership they have going on. An example of this is there's a fungi called mycorrhizal, and it attaches to plant roots and helps plants absorb water and nutrients. And in return, that plant shares their sugars with the fungi. So fungi come in all different forms. There's yeast, which we use to like make bread and beer. There's mold, which is a fuzzy black mold that you tend to see on like old bread. And then we also have mushrooms, which are simply the fruiting bodies of fungi. Some are edible, like oyster mushrooms, and others are toxic, like the bright red fly arctic mushroom. Okay, so characteristics of fungi. Fungi are really unique because of their cell walls and their membranes. Their cell walls contain chitin. It's the same material in insect exoskeletons, so in a way, mushrooms and cockroaches have something in common. This makes fungal walls tougher than plant cell walls. They also contain glucans, which are polymers of glucose. These are really important for some of the antifungal drugs that we use because it helps to target the glucan synthesis. Instead of cholesterol, fungi have ergosterol in their membranes. This is what makes the antifungal drug possible. An example would be is if somebody had oral thrush, which is a yeast infection caused by candida, they might be prescribed fluconazole, and this drug targets the ergosterol, weakening the fungal membrane until the cell dies. So fungal structure. So most fungi grow as molds, but some grow only as yeast. Certain species are dimorphic fungi, meaning they can switch between yeast and mold forms depending on the environment. So an example is there is a mold that's called Histoplasma capsulatum, and it grows as a mold in the soil, but when it infects humans, it switches to a yeast form. Molds grow as long filaments called a hyphae. A visible mass of hyphae is called a mycelium, which is what you actually see when mold spreads on old fruit or bread. Many fungi have a septate hyphae. Septa are cross walls with pores that let the cytoplasm and the nutrients flow through, almost like rooms in a house with open doors. Other fungi have an aseptate hyphae, long tubes without walls. So they look like one giant cell with many nuclei floating inside. A common or an example of that would just be like the common bread mold, rhizopus. Fungi are not motile, so they cannot move around like bacteria. Instead, they grow at the tips of their hyphae, spreading toward food sources. Some hyphae grow upward as an aerial hyphae, which produces spores. These spores are what help fungi reproduce and spread. The fuzzy or powdery look on moldy food, that's actually the aerial hyphae, and it's covered in spores.
So fungi reproduce both asexually and sexually. Most commonly in lab cultures, we observe asexual reproduction, which includes spore types like conidia, arthroconidia, and sporangiospores. Conidia are spores formed on conidiophores, while arthroconidia are formed by fragmentation of hyphae. Sporangiospores form inside a sac called the sporangium, and yeast reproduce asexually through budding, which is where the nucleus divides by mitosis and goes into a daughter cell that pinches off. These structures are crucial in medical identification because they're visible under a microscope in culture. Sexual reproduction involves two different mating types, which are labeled plus and minus, whose hyphae fuse. This results in cells with two haploid nuclei, which may eventually fuse and undergo meiosis, which forms sexual spores. These sexual forms help with fungal classification, though they're not always seen in lab settings. A visible example of a sexual structure would be a mushroom, which is the fruiting body of some fungi. Fungi can infect human health in several ways. We have mycoses, also known as fungal infections, and they are classified by how deep they affect the body. We have superficial fungi. These infect keratinized tissues like skin, hair, and nails. Examples include dermatophytes, which cause conditions like athlete's foot and jock itch, and Malassezia furfur, which causes a skin discoloration called tinea versicolor. We also have subcutaneous fungi. These infect deeper skin layers, often through trauma. For example, we have Sporothrix schenkii, which causes a disease called sporotrichosis, also known as rose gardener's disease. Opportunistic fungi cause disease only under certain conditions, mainly in immunocompromised people. These can include Candida albicans, Pneumocystis gyrovecii, Aspergillus, Cryptococcus, and Rhizopus. Systemic fungi, like Histoplasma capsulatum or Coccidioides immitis, can infect even healthy people. They're often dimorphic, meaning they grow as molds in the environment, but they convert to yeast forms in the body. Fungi also produce dangerous toxins. Aspergillus produces aflatoxins, and these are carcinogenic and often found in grains and peanuts. Claviceps purpurea causes ergot toxin, which is a hallucinogenic and also the source of the drug ergotamine, which is used to treat migraines. Amanita species, especially Amanita phalloides, produce deadly liver toxins. And finally, mold spores can act as allergens. They often trigger asthma or allergic reactions in sensitive individuals, which is why mold counts are often included in weather reports.
Hi guys, my name is Ava Rosen, and I'm going to be going over how we can apply the Christian worldview to the study of microbiology. Human dignity and value. A Bible verse that reflects the Christian worldview of human dignity and value is Genesis 1:27. So God created mankind in his own image. In the image of God, he created them. Male and female, he created them. The Bible verse highlights that we are all made in his image with great purpose and value, and to live a life of service to him. We can honor God by treating everyone, including patients, coworkers, and other healthcare workers, with respect, gratitude, and kindness, thereby improving the health and well-being of God's creation. Ethical practice to the common good. We can apply the Christian worldview to the identification and classification of microorganisms in their diversity by recognizing how purposeful God is with his creation and how he equipped those with skills to serve others. The study of microbiology gives us the ability to deepen our understanding of God's creation at the biological level, which demonstrates God's purpose of creation. Deepening our microbiological understanding is an act of appreciation for the Lord and his beautiful creation, and also a form of protection from sickness and the spread of disease. Through utilizing the skills to better the health of others, we serve the Lord by caring for others with honesty and compassion, human flourishing, and the common good. Mark 12:31 states, "The second is this, love your neighbor as yourself. There is no commandment greater than these." Microbiological studies and discoveries have led to life-saving treatments, vaccinations, and prevention methods that promote better and healthier lives. Through completing studies, making discoveries, working in the healthcare industry, and helping patients, we are honoring the Lord and his commandment of loving others, which promotes human flourishing and the common good.